Hello YouTube. I thought I'd make a video today talking about glycogenesis, glyconeogenesis, and glycogenolysis. These are hard words to say, but anyways, the reason why I'm making this video is because I thought I could offer a better explanation to people about how these things work, because these things are very important for understanding metabolism, and my original understanding wasn't very good, and I think I can make a better video than most of the people because I can expand and explain how this works like within a very broad picture and relate it to other ideas that are important to just generally understand. And the way I explain things will help you to actually get these things down rather than just rote memorizing them. It'll help you to understand trends and help you to understand like the meaning behind the words within these enzymes. And so I'm going to explicate this in this video. So let me get into it right now. So starting off with glycogenesis. Probably going to butcher this a couple times in the video. This occurs because we have high blood glucose, right? When we're in a state where we don't need to rely upon, the Cori cycle and lactate and things like this, when we have high levels of blood glucose, our body will store it, right? Because oftentimes humans, throughout our history, went through periods of extreme starvation and then periods of, eating a lot of food. And so we came up with inventing glycogen, or we didn't come up with it, we evolved it, similar to how other plants use things like cellulose to store energy, to store carbohydrate chains. And so this is kind of our version of cellulose. So a lot of animals or just things within life, organisms, use this method to basically store energy, because oftentimes nutrients and getting energy was very ephemeral, and it didn't last long, so we need to store this. So now really getting into this, just explain why we do this is because we have periods of starvation, we store glycogen for those periods, and then we have gluconeogenesis, which comes after. So when, let's say, we're in a period where we just got out of a big fast, we've been fasting for 24 hours, all of our glycogen stores have been depleted, and we're now trying to build it back up because we just had a lot of rice. What happens firstly is high glucose will go into the liver, where it is then turned into glycogen. This is basically a broad picture scheme. How this actually happens is when insulin binds to the GLUT receptors on muscle, and particularly liver, it basically causes these GLUT receptors to translocate to the cell membrane and allow glucose to enter the cell, where it will then be turned into glycogen through a series of chemical processes. Now, getting into the first step, we take glucose into the cell using ATP. Once we take glucose in the cells, we have to turn glucose into glucose 6-phosphate. If you understand glycolysis, you'll understand glucose 6-phosphate using hexokinase is the first step. And this is a step that requires energy, along with the third and the 10th step, those are actually irreversible steps. But yeah, so once we take glucose, we're going to use hexokinase to facilitate this reaction to get glucose 6-phosphate. Glucose 6-phosphate is essentially glucose with a phosphate on the 6th carbon. And I'm explaining this kind of to help people understand what the structure of it would look like. Here I have a normal glucose molecule, and on the 6th carbon, so it'd be 1, 2, 3, 4, 5, 6. Well, 5, we'd have CH2, and then a 6-phosphate over here. That would be what our glucose 6-phosphate would look like. Then, once we have glucose 6-phosphate, we're going to have to turn this into a more stable form. Glucose 6-phosphate is highly reactive and will get used up in glycolysis, right, to get turned into fructose 6-phosphate. And so in order to turn it into a more stable form that can be used for later energy usage, we have to create glucose 1-phosphate. And I've noted this here as it being very stable. This is done using phosphoglucose mutase, the enzyme using phosphoglucose mutase. Think about the reasoning, or think about the nomenclature, the semantics being used here. Mutase is an enzyme that will mutate something, right? It's essentially the same thing, but it's just in a different form. It's kind of similar to how isomerases work, right? So we take glucose 6-phosphate and turn it into glucose 1-phosphate. This is a stable form. Then, this is a pretty important step. We use UTP, uridine triphosphate, to turn it into UDP glucose. This will be very important in later steps, and I'll explain why. This is done by a pretty difficult to pronounce enzyme known as UDP6 pyrophosphorylase. Pyro basically meaning six carbon chains, and a phosphorylase is an enzyme that basically cleaves bonds using the addition of phosphates. I really wish my hand was more steady right now. Anyways, Phosphorylases will cleave bonds adding phosphates. Phosphatases will cleave bonds by removing phosphates. So you should probably write that down. I think I actually listed this at the bottom, so I'll restate it again later. This is an important step to understand. Once we create this UDP glucose, we need to create chains. How do we do this? We have to create 4 UDP glucoses to actually start the chain. We use an enzyme called glycogenin to help kind of kickstart this to collect these UDP glucoses. So imagine UDP glucose being added together in a chain, and it's being started by the sort of catalyst known as glycogenin. Glycogenin will basically just allow glycogen synthase, which is the enzyme that will basically continue adding glucose molecules onto this chain, or UDP glucose molecules onto this chain, to create glycosidic bonds. I noted ether bonds. The reason why I note this is it's just important to understand this from an organic chemistry perspective, where everything is very microscopic, very down to the level of single bonds. This is an ether bond, right? A good mnemonic to understand ether bond is basically ROR, which would make you say or. And a funny mnemonic I used to use to help me to memorize this is just you either get it or you don't. And then when I write it out, I'll just say OR to help me understand that ether is that. And so these are glycosidic ether bonds. And I want you guys to understand this because it's going to help you to understand the chemical changes that occur with the functional groups when we catalyze these reactions. Then, once we create these, well, actually, before we create these ether bonds, I'm going to continue talking about why we use UDP glucose. UDP glucose is created because in order to, put things in a lower energy state, right, or to create chains, you're actually increasing entropy, which is unfavorable. You have to use energy to catalyze this reaction. And so the UDP is essentially a leaving group, almost a bit like an SN2 reaction, which will provide the energy for the conversion of an ester bond to an ether. And so initially we have these ester bonds, which is the UDP glucose in its original form, and we're going to turn that into ether bonds. I will probably provide some later discussion on this as well. Now I provided a graphic to explain what glycosidic bonds look like and how they form. Understand that this is oh my god, man, I wish my hand was steadier. Understand that this is a condensation reaction where we basically take an OH groups in alcohol and one hydrogen off of an OH to form H2O. So it's a dehydration reaction, forming an ether. Pay attention, there's a hydrogen, and the camera isn't picking it up because it's very small, but there's an oxygen. And that hydrogen stays sticking out, but this oxygen just forms directly to this carbon, forming a glycogen bond. This is catalyzed by glycogen synthase. So imagine glycogen synthase just catalyzing this. And throughout this whole time, UDP glucose, the UDP part, as glycogen synthase is catalyzing this, the UDP is leaving. The UDP that was once attached is leaving from that phosphate group, right, from this ester to an ether. The UDP is leaving, and it's leaving, and it's leaving, and it's going to help create these chains. Now, an issue that we run into is that this is linear. And when we want to store glycogen, we don't want to store it in a linear fashion because it takes up a lot of space. It's not very sterically, so to say, favorable. We want to increase our surface area and we want to make this very dense. So we use something called a branching enzyme. Branching enzyme, what it does is it basically cleaves a 6-member segment of the bond that we created, of like from carbons 1 to 4 to create 1,6 bonds. So what this means is carbons 1, 4 are connected by an ether. So if I count for you real quick, this would be 1, 2, 3, 4. So this is the first carbon, and on the second carbon, 1, 2, 3, 4. They're connected at the first and the second carbon. But then we're going to take this and we're going to convert it into the second carbon. And I drew a little graph to help explain this. So imagine this. This is the glucose, this is the ether bond. So 1, 2, 3, 4 glucoses, and they're all connected by ether bonds. Well, branching enzyme is basically going to take part of this and almost kind of chop it off, so to speak, and reconnect it at the 6th position. Think about this. Like I said, this is 1, 2, 3, 4, 5, 6. Or 6, I'm sorry, here, because it's CH2 group up here, CH2OH. And we're going to connect it here. And so we're going to take one of the glucoses and we're going to connect it at the 6th position, which will be up here. And that's what it essentially looked like. hopefully this does make sense to people. Now, I wrote down here because I want to reemphasize this. Phosphate groups or phosphatases, right? Glucose phosphatase will cleave bonds by removing a phosphate, which forms an ether, right? Phosphorylases cleaves bonds by adding phosphates, which forms an ester, right? So if I'm adding a phosphate group, I have an ester bond here. The camera is not picking it up. I have an ester bond right here. Honestly, I should have used better handwriting, but hopefully this makes sense. Now that I just explained this, I'm going to give a quick overview recap of all of this. What we do is we take glucose, convert it to glucose 6-phosphate. Glucose 6-phosphate is acted upon by phosphoglucose mutase to turn into G1P, which is more stable. G1P is then acted upon by UDP6 pyrophosphorylase, which will once again cleave bonds by adding a UDP. The UDP is used as a leaving group to help grow this chain, and that is grown by glycogen synthase, and it's started by glycogenin. Now, because this growing chain gets really long, we need to use branching enzyme to cut these 1,4 bonds to form 1,6 bonds, and that's essentially glycogenesis. Now, getting to glycogenolysis. Glycogenolysis is pretty much the reverse of this, but has a couple of other extra added steps. Firstly, what we do is once we have the glycogen molecule, let's say we're in a state of starvation. So what happens is epinephrine or glucagon gets released and it attaches to ligands, which would be, I'm pretty sure it's a GPCR. Yeah, it's a GPCR, which will cause a cascade reaction to cause this. And I'm going to explain these steps later as well. What happens initially is glycogen phosphorylase is going to be used to cleave the linear strands to create G1P. Remember, the first step we had, or the second step we had, was the creation of G1P. So the individual monomers are G1Ps, not G6Ps. So we're going to have G1Ps, and pay attention to this because I made sure to be really important in how I drew this out. We're going to have 1, 2, 3, right? 1, 2, 3. And we're going to use a phosphorylase to cleave off one of them to create a free G1P. So we took off one of the glucoses, and now it's G1P, and then that is I'm drawing the system symbolize where the PO4 will now be at the first position, right? Now, getting into another enzyme. Well, since we put this glycogen in a branched form, we actually now need to debranch it because glycogen phosphorylase can only act on the linear form. So we use this enzyme called debranching enzyme to basically remove the branches and to create a linear chain, to relinearize it. And this is composed of basically two sub-enzymes, 4-alpha gluconeogenesis and alpha-1,6 glucosidase. 4-alpha gluconeogenesis, if you think about that, transferase, it's going to transfer things, right? And it's going to be alpha, meaning the anomeric carbon is in the alpha position, and then 4 meaning the fourth, at the fourth carbon. So let's say we have a branch here. We're going to 4-alpha gluconeogenesis is going to take this branch part at the 1,6 position and reconnect one of them to form a bond at the 1,4 position. I actually should have drawn this here. It's actually this. Notice how I only have two, what's it called? Carbons, or not carbons, glucose molecules. Now I have 3, but notice I intentionally left one up here because we have a free glucose G1P sitting up here that's just stuck there. Well, debranching enzyme must only leave one. Well, how do we get rid of this free one? Well, this is where alpha-1,6 glucosidase comes in. It takes that lone glucose, that lone G1P, and it's just going to create a free glucose. It's actually not even a G1P, it's just a free glucose. So it's going to take this glucose sitting up here and it's just going to give us a free glucose. So an important thing to understand is that debranching enzyme will take 3 branch pairs, three carbons, and add it to 1, leaving 1. And in the process, it creates 1 free glucose, which can be used to free up energy. Now, since we have all this G1P, right, that we're liberating, it's in its stable form. We now need to make it, in its reactive form, which is G6P, which the body can use. Think about this, phosphoglucose mutase. Remember, that is the same enzyme we use in glucogenesis. Phosphoglucose mutase is what turns G6P into G1P. phosphoglucose mutase can do the reverse of this as well, which is going to turn G1P into G6P. This G6P can then be used in glycolysis. The thing is, though, is that our muscles, when we're exercising in fasted state, cannot direct. They can use G6P, but the thing is that in a fasted state, G6P cannot travel through the blood. It has to be in its stable form, which is glucose. And so what we're going to do is we're going to turn this G6P back into glucose. This seems a bit redundant, like we're, going reverse just to go back to where we were, but biochemistry is very redundant, and we have a lot of these chemical processes in the body. So just be aware of that we're actually going to you know, a product of glucose, which is using glycolysis, but we're turning it back into the product. We're going in reverse, only to go back forward. Then glucose 6 phosphate, right? Glucose 6 phosphatase, right? Once you have glucose 6 phosphatase, in order to get it to glucose, we have to cleave the phosphate from the 6 carbon. So that's where glucose 6 phosphatase comes, right? We're taking away the phosphate to 6 carbon, and we're going to get rid of it to just turn it into glucose, just bare bones glucose. And so I've drawn a little wording here to say G6P gets turned to glucose, right, by glucose 6 phosphatase, which goes in the blood and then can be used by muscles for glycolysis. Now, I just drew quick overview here. Essentially, in glucogenesis, glucose gets turned to G6P by hexokinase. G6P gets turned to G1P by mutase. This is the stable form. Then G1P is basically stacked together using 1,4 bonds by glycogen synthase. Glycogenolysis, basically glycogen does reverse. We phosphorylate to free G1Ps, right? We're breaking bonds, adding phosphates. So we have G1P, and then we're breaking the 1,4 bonds by using phosphorylase to create free G1Ps, and debranching enzyme is going to linearize the chain while also creating one free glucose. Now, let me get into this. Here's a broad picture overview of what's happening here now. This is where I think this video becomes very helpful. In glucogenesis, right, when we're in a high energy state, our blood glucose is high, insulin then gets released by the beta cells of the pancreas. What happens is insulin, as I've drawn here, attaches to this little ligand. Now, I could have been a bit more detailed in this, but I just drew this, RTK. I can make a video about RTKs, but essentially what happens is RTKs they respond to peptide hormones. Insulin is a peptide hormone secreted by the islet cells, and the RTK will basically it's basically a homodimer, and then it'll transphosphorylate itself, leading to a cascade. What happens is this basically liberates the phosphate from glycogen synthase, enabling it to be active. Now, think about this. This is a bit counterintuitive. Typically, we think about kinases activating things, or once you attach a phosphate, it's activated. For glycogen synthase, it's reversed. For glycogen phosphorylase, this is the case, where adding a phosphate is activating it. Glycogen phosphorylase is basically the antagonist enzyme to glycogen synthase. Think about that. Glycogen synthase is inactive when phosphorylated. Glycogen phosphorylase is inactive when non-phosphorylated, but active when phosphorylated. So the only difference between glycogenesis and glycogenolysis is the addition of a phosphate. And so this is ultimately going to cause, the ligand binding to the RTK is going to liberate this and cause a phosphate to be added to remove a phosphate to add to glycogen synthase. Now, glycogenolysis. This, because it's responsive to glucagon, which is secreted by the alpha cells of the pancreas, it's going to bind to the GPCR. GPCR is basically a 7-membered ring, and it says G because it kind of looks like it forms, G's. It's in and out, and it's a 7-membered, I guess, receptor that once a ligand binds to it, it initiates a cascade. The first cascade is the alpha subunit will dissociate from the beta and the gamma subunit. I haven't drawn them here because I'm assuming that people in this video kind of already know how this works. The alpha subunit then will go and bind to another molecule called adenylyl cyclase. Adenylyl cyclase is basically an enzyme that will catalyze that will turn a substrate into a cyclase form, right, as it's in its name. So adenylate cyclase. You can also have guanylate cyclase, which turn GTP into cyclic GMP, but it's adenylate. So for adenine, the adenylate cyclase is going to take ATP in the cell and turn into cyclic AMP. Cyclic AMP is in the secondary messenger molecule, which is going to go then and bind to pKa. pKa will then add a phosphorylase to add a phosphate to the phosphorylase, which will then make it active, right? And so let me get into the discussion here. pKa phosphorylates downstream targets to activate glycogen phosphorylase and deactivate glycogen synthase. So once it binds to GPCR, a phosphate is added to both phosphorylase and synthase. But if a phosphate is added to synthase, it's inactive. And if a phosphate is added to the phosphorylase, it's active. So that's an important thing to understand in all of this. Now, this video is already getting really long. It sounds better at making these. But now imagine when we've exhausted all of our glycogen. We get in a state where, we've ran out of glycogen, we've been starving for way too long, and we need to make energy from something new. Think about the name, gluconeogenesis. Neo means new, genesis means creation, glucose. So we're creating glucose from new sources. And this can come in the form of lactate, can come from the form of amino acids, can come in the form of fatty acids using glycerol. And I'm going to explain this now. Gluconeogenesis is basically making glucose from new forms. And it basically is the reverse steps of glycolysis. But the important thing to understand is that we have to work our way around these irreversible steps. And this is important. It is the first step, the third step, and the 10th step. So the first step, we go from glucose to G6P. The third step, we go from fructose 6-phosphate to fructose 1,6-bisphosphate. And the 10th step, final step, phosphatidylpyruvate to pyruvate, which is what's used in the Krebs cycle. This is very important to know because phosphofructose 1, phosphofructose 1, phosphofructose kinase 1 is the rate-limiting step in glycolysis. This is because it's highly regulated by things like AMP and citrate levels. And this is just an important thing that you should understand when it comes to glycolysis. So if we have high levels of ATP in the cells, or we have high levels of citrate in the cells, we're actually going to inhibit phosphofructose kinase 1. But if we have high levels of AMP, which is ATP without 2 phosphates, we're going to increase the function of phosphofructose kinase 1. Now we're going to work our way around this using 4 enzymes: pyruvate carboxylase, phosphoenolpyruvate carboxykinase, phosphofructose 1-bisphosphatase, glucose 6-phosphatase. These bottom 2 enzymes are pretty intuitive to understand, but the top 2 require a bit of deliberation to get down. But I've made sure to help explain this from an organic chemistry standpoint. So now imagine we have pyruvate, right? Pyruvate is the final step or the final product of glycolysis. We need to turn pyruvate back into glucose. So how do we do this? We are going to basically have to send it back into the Krebs cycle. If you understand the Krebs cycle, you'll understand that we go from fumarate to malate to oxaloacetate. Oxaloacetate is the final product of the Krebs cycle where it has to be combined back with acetyl-CoA to form citrate. So oxaloacetate, I've drawn it here. Pay attention. Well, actually, before I say that, pay attention to pyruvate. Notice that we have basically ketones. These are ketones and alcohol. If you look at the name pyruvate carboxylase, you can probably imply that we're going to do something with a carboxylic acid group, right? And that's exactly what happens. We're basically just making this longer by adding carboxylases to these ends. So we're moving this alcohol and adding carboxylase and adding carboxylase here. Also know that we go from a 1, 2, 3 chain, carbon chain to a 4. 1, 2, 3, 4. Just note that. Now, this is going to get a bit tricky, but bear with me here. Once we go back into the mitochondria, right? Oxaloacetate cannot leave the mitochondria because it's charged, because it has these carboxylic acid groups. But guess what isn't charged? It's precursor, malate. Like I said, in biology, we have to move a step back just to go back forward. And that's what we're doing here. We go from oxaloacetate to malate, and this happens via an oxidation reaction, right? And so that means that we're taking NAD+, right? And we're turning it into NADH. Oxidation is going to turn an alcohol to a ketone. I'm going to say it again. Oxidation is going to turn an alcohol into a ketone. Very important to understand. Malate is a ketone. Oxaloacetate has an alcohol on it. The reverse is true. Reduction will turn a ketone, reduction, adding electrons, right, which comes in the form of hydrogen, will turn a ketone into an alcohol. And we use this using NAD+. So this is oxaloacetate, and this is malate. Malate is a ketone, so we oxidize an alcohol to go to a ketone. Then malate is free to leave, and once it leaves the mitochondria, it then spontaneously gets turned into oxaloacetate and gets reduced in the cytoplasm. So we could use oxaloacetate, but it can't leave. So we have to turn it into a precursor. Then once the precursor leaves, once it's out, then it can be converted to oxaloacetate. Oxaloacetate then gets acted upon by phosphoenol pyruvate carboxykinase. And this is catalyzed by turning GTP into GDP. And that turns into PEP. PEP is a step right before what's it called? Pyruvate. Which is created by pyruvate kinase. Then we are going to go through the steps of turning pyruvate into 2-phosphoglycerate using enolase, 3-phosphoglycerate using mutase, 1,3-bisphosphoglycerate, and then we're going to get to fructose 6-phosphate using aldolase. I didn't draw these out because what I just said is the exact reverse step of glycolysis. But once we get to fructose 1,6-bisphosphate, right, all we have to do is just turn it into its precursor, which came before it, which is fructose 6-phosphate. So think about this. Fructose 1,6-bisphosphate. Bis means 2. There's a phosphate at the first and a 6 carbon fructose. But then here we have just a phosphate at the 6 carbon. So that means we must have done what? Taking away a phosphate. So think about the name. We're using fructose 1,6-bisphosphatase. And I've drawn it here, bisphosphatase. This camera just does not pick it on my handwriting. Then, once we have that, we have fructose 6-phosphate. Then, from fructose 6-phosphate, that gets turned into glucose 6-phosphate by phosphoglucose isomerase. Once again, glucose and fructose are isomers of each other. Then we have glucose 6-phosphate. Just like we did in this step, we're just going to use a phosphatase, glucose 6-phosphate, to turn it into glucose, and this is glucose. Instead of an OH group, we're going to have an ester bond with a phosphate. And that is glucose. That is how we do gluconeogenesis. But you might ask, okay, but where did the pyruvate come from in the first place? Now I'm going to explain that. So let's say we have intermediates. Lactate, for example, which is generated during exercise, can be used in the Cori cycle. So what we're going to have is lactate, which is this structure, is going to be acted upon by lactate dehydrogenase. Understand that a dehydration reaction, a dehydrogenase, which is going to remove an OH, right, which would then turn into a ketone, is oxidation. We're oxidizing it. We're reducing the electrons here, right? Or I shouldn't say reducing because you make think reduction, but that's essentially what it is. When you say reducing, you're basically reducing its, I think, electron number or something like that, or its charge, essentially. So you go from an alcohol to a ketone. Once again, oxidation will turn alcohol into a ketone. This is pyruvate. Fatty acids can also be used. Triglycerides, which is basically this, but with 3 glycerol molecules coming off of it, is that. And we're going to take the glycerol used from fatty acids and act upon it using glycerol kinase. Well, if you think about that, glycerol kinase, well, kinase is add phosphate. So we're going to add a kinase somewhere. Well, we're actually going to add it to the 3rd carbon, and that is glycerol 3-phosphate. Glycerol 3-phosphate is going to then be used by glycerol 3-phosphate dehydrogenase, right? Once again, we're going to oxidize this, take one of these, alcohols and turn it into a ketone, which is DHAP. If you understand metabolism, you understand DHAP is a product of aldolase acting upon fructose 1,6-bisphosphate. DHAP and glyceraldehyde 3-phosphate are used. Then TPI will convert DHAP to glyceraldehyde 3-phosphate, which is then able to be used to create glucose. Now, amino acids. Just understand this. All amino acids except leucine and lysine are turned to malate, which then, once again, talking about from the previous steps we used, will be converted right back into glucose. I'm going to cut this video short. I was going to have a discussion, but I'm just going to restate this. Oxidizing alcohol turns into a ketone. Reducing ketone turns into alcohol. Think about this. This is a ketone. I'm going to reduce it. So I'm going to take a hydrogen off and I'm going to turn it into alcohol. I'm reducing it. Hydrogenation is reducing. Dehydrogenation is oxidizing. Hopefully you guys found this video helpful. I don't think it's my best video because this is really hard to get through. My handwriting was bad. Camera wasn't really picking everything up, but I hope you guys understand these trends. Thank you.